I am Sri Jaisavitya and I am going to present on the topic metagenomics. Metagenomics is the study of all genomes of organisms isolated from a bulk sample. It is particularly useful for studying communities of microorganisms in various environments such as human skin or water samples. It is an interdisciplinary field of genetics. It includes cutting-edge, futuristic, state-of-art and high-throughput sequencing analysis for microbial studies. It focuses on community genes rather than individual organisms. The metagenomics addresses the limitation of traditional microbiological techniques where most of the microorganisms cannot be used cultured under standard laboratory techniques. So it is culture independent and is used to study the vast majority of microbes that are unculturable or difficult to culture. Traditional cultivation and culture-based microbiology techniques are inefficient for exploring the uncovered and unknown portion of the present plethora, which makes it particularly impossible to identify every microbe present in the system. Another problem is that with new age 16S rRNA gene sequencing, we can only determine the taxonomic information and for high abundance microbes only. Yet again, a high concentration of the microbial community and less abundant sequence remain unstudied. Metagenomics solves both these critical problems and allows us to investigate microbiomes effectively with speed, accuracy and precision. It allows the exploration of microbial diversity, including unculturable microbes, which represent more than 99% of the microbial life. Provides insight into microbial community structure, function and interaction in various environments such as soil, water, human gut and extreme habitats. The development of molecular techniques such as the use of 16S ribosomal RNA sequencing allowed the study of microbial communities without the need for cultivation. This built the foundation of metagenomics. Joe Handelsman and colleagues coined the term metagenomics in 1998 and defined it as the study of collective genomes of microorganisms in environmental samples. The early 2000s saw the development of high-throughput sequencing technologies which allowed the rapid sequencing of entire microbial communities. Now, let us take a look into the history of DNA sequencing. The Sanger sequencing method was developed in the year 1977. The human mitochondrial genome sequencing was done in 1981. The Human Genome Project launched in the year 1990 and got completed in the year 2003. The complete eukaryotic genome was sequenced in the year 1996. The first NGS technology was released in the year 2005. The Human Microbiome Project was launched in the year 2007 and it completed in the year 2019. The PacBio sequencer released in the year 2011. Principle of metagenomics. Principle of metagenomics involves studying the genome of microbial communities in bulk environmental samples directly without culturing individual species. This allows detailed analysis of microbial communities and overcomes the limitation of traditional laboratory culture methods. The process in metagenomics includes collecting environmental samples, extracting DNA from the microbes present in the sample, sequencing using high throughput technologies and analyzing the sequencing data with bioinformatic tools to identify and characterize the microbial communities in their natural habitat. It is performed using two main methods. The first is targeted sequencing, which is based on the principle that targeting specific regions of an organism's genome can be used to identify and characterize the organism. The second is shotgun sequencing. Its principle basically is based on the random sequencing of all the DNA in the sample. It provides a detailed analysis of the genetic material from all organisms in the sample. Objective of metagenomics: identify microbial diversity, discover novel genes and enzymes, understand microbial functions by studying pathways and ecological roles of these microbes, analyze microbial interactions and its application in various fields include improving medicine, agriculture, industry and environmental management. There are two types of metagenomics. One, targeted metagenomics and the second one is shotgun metagenomics. Targeted metagenomics is also known as amplicon-based sequencing, which involves sequencing specific genetic marker or conserved regions within the microbial community such as 16S rRNA for bacteria and 18S rRNA or ITS regions for fungi. These regions have variable sequences that are useful for identification of different organisms present in the environmental sample. This method generally does not provide strain level resolution. Shotgun metagenomics or whole genome shotgun metagenomics involves random sequencing of all genetic material in the sample. Unlike the previous method, it focuses on sequencing all the DNAs present in the sample. This provides detailed information on both the taxonomic and functional composition of the microbial community. It also allows species level identification. The basic process of metagenomics involves sample collection, DNA isolation, DNA library preparation, DNA sequencing, and analyzing the sequencing and reconstruction of the sequences, assembled genome sequences, and tools and software and analysis. This slide provides detailed information of how the metagenomics works. The first step is sample collection, second DNA isolation, third library preparation. After the preparation of library, we are sequencing, then the metagenomics assembly. The assembled metagenomics is then processed to gene prediction, then annotation, then pathway prediction, and comparative metagenomics analysis. Now let us look deep into the steps involved in metagenomic analysis. The first step, sample preparation. Proper sample preparation, preservation and processing are important to maintain the integrity of genetic material and ensure the accuracy. The process starts with the collection of environmental or biological samples. Second step, DNA extraction. Once collected, the samples undergo processing to extract DNA. Different physical and chemical methods are used for DNA extraction depending on the sample type, the site quality and quantity of DNA. The cells are lysed to release the DNA and the cellular debris is removed using centrifugation or filtration. The DNA is purified using methods like phenolchloroform extraction and silicocolumn based kits. The extracted DNA is then stored at appropriate temperatures. The sample types include soil, water, gut microbiome and extreme conditions like hot springs and deep sea. The DNA extraction method includes mechanical, chemical and enzymatic lysis. The mechanical methods are beating and grinding, cell lysis, detergent buffers, enzymatic lysis, lysosome and protease K. DNA sequencing. The third step is DNA sequencing. It is a key step here. Sequencing reads the nucleotide sequences present in the sample. The Sanger sequencing and various NGS platforms both can be used. Depending on the platform, DNA is either fragmented into smaller pieces, cloned into a VAC vector or directly employed for sequencing. The end result of any sequence would be a file containing nucleotide sequence, many short, long and good and bad reads. Common sequencing platform includes traditional Sanger sequencing, Pyro sequencing, Illumina system, solid sequencing by Applied Biosystem, HiSeq 2000 by Illumina, single molecule sequencing by PacBio, ion torrent and ion proton sequencing. The fourth step is assembly. It is a process to collect, arrange and reconstruct reads to complete the sequence. Various computational tools are available now for different assembly process and complete metagenomic analysis. The two commonly used assembly options are de novo assembly and co-assembly. The de novo assembly is performed when a set of reference sequences or reference genomes aren't available to compare and arrange. Hence, this process is tedious, complex, time-consuming and requires immense computational power. Whereas co-assembly is a reference-based assembly process, the target genome is compared with the reference genome or sequence available in the database. It's a comparatively less difficult process but needs high-end computer tools for analysis. The fifth process is binning. It is the process of grouping or sorting assembled DNA sequence into groups or bins that represent individual genomes from closely related organisms in a microbial community. Various algorithms have been developed for binning that uses different types of informations combined within the DNA sequence. Computational based binning algorithms use the conserved nucleotide composition of genomes. They analyze sequence fragments for patterns in nucleotide composition such as GC content to group them into bins. Similarity-based binning algorithms rely on the similarity of DNA fragments to know genes in reference databases. Some binning algorithms combine both compositional and similarity based methods. They combine information on nucleotide composition with sequence similarity to improve binning accuracy. The sixth step is gene prediction. Binning gives us a rough idea about what genomes or sequence we have and where they belong. The next step allows prediction of potential genomes present in the assembly, thereby protein produced by each if any. Eventually, it gives us a broad idea about the function of the entire metagenome. This means, depending upon the type of presence of genes, what type, number of functions a particular community performs in the system, environment or ecosystem can be determined. The seventh step is gene annotation. The genes within the assembled sequence are identified and annotated to predict their functions using bioinformatic tools and databases. It involves two main steps, feature prediction and functional annotation. Feature prediction identifies genes or other genomic elements present within the DNA sequence. It also identifies non-coding RNA, regulatory sequences, binding sites, structural motifs, and other biologically relevant elements. In functional annotation, it identifies function of the predicted gene. Then, the eighth step, experimental design and statistical analysis. Experimental design and statistical analysis are important in metagenomic studies to ensure valid and interpretable results. Effective statistical method interprets the vast amounts of metagenomic data generated and handle the complexity of the DNA data. Various statistical tools are used to analyze metagenomic data. The primary package and web-based tools like Metastats are multivariate statistical analysis. This picture represents how the metagenomic works. Advantages of metagenomics. It is culture independent, simultaneously identifies taxonomized genes and functions. It enables discovery of new genes, enzymes, antibiotics and bioactive components. It also reveals microbial roles in nutrient cycle and ecosystem balance. It helps understand the human microbiome and its link to health and disease. It detects microbial changes in polluted or changing ecosystems. Disadvantages of metagenomics. It is high cost, complex data analysis requires advanced bioinformatic tools and expertise. Many genes cannot be matched to known functions. Sample handling may introduce external DNAs. DNA extraction and sequencing may favor certain organisms, which results in biased representation. It is difficult to link genes to specific microbial species or function, which includes interpretation challenges. Applications of metagenomics. It is used to study human microbiome. It includes research on gut microbiota, skin microbiota and their roles in human health and diseases. Used to identify pathogens in clinical samples. Understanding the microbial diversity and functions. Monitor water quality and detect pollution. It is used in bioremediation and it also helps us in understanding the microbial communities associated with plants, which can improve crop health and productivity. It helps us identify microorganisms that are capable of energy production. It also is useful in discovering new enzymes, bioactive compounds, antibiotics, and chemicals. Thank you.